Thank you, Professor Wong, and thank you all of you for coming. And it is my great honor to deliver this Professor Ren Guorong lecture. Today, I'm going to share with you the recent work. Actually, most of the work I'm talking about were done during the pandemic period. So this is conventional structural biology, the structure of a protein. Actually, I don't know which one, but we know it's a protein structure. So this is a snapshot. So many of our colleagues work hard to get structures of a protein. But this structure is like this. Okay. It's a nice picture of a symphony orchestra, but you don't know, you can't hear what they are playing. The function of a protein, we consider, for example, the orchestra is like a protein, and every player, every instrument is an amino acid residue in the protein. But the protein function is dynamic, and the function of protein is a process. So in order to know, to correlate the structure with the function of a protein, we actually have to do a time-dependent structure. Okay. And what is shown on this slide is just an example of an enzyme catalysis. So we all know this is a simple enzyme, a common enzyme, DNA polymerase, which catalyzes the replication of DNA. But catalysis is not a single-step reaction. Okay, even though in the textbook we often consider it as a single step, the enzyme started as a free enzyme, then it has to bind DNA, it has to bind DNTP nucleotide, and there are 4 different nucleotides. It has to match with DNA, and then the enzyme then undergo a conformational change, then has to bind metal ions or more than one metal ion, goes through transition state, and then give the product, and then the product has to get released, going through multiple steps. So actually, in the field of DNA polymerase, there are thousands of structures solved already, reported. If we go to the protein data bank, thousands of structures from different DNA polymerases, from different species. They are not all the same because researchers are trying to catch different intermediates in order to understand the process. So some of them may work on the free enzyme, some of them may work on the enzyme DNA complex, some of them may be on the enzyme DNA and nucleotide complex and protein complex and so on. Now, in order to catch these intermediates, researchers often have to use analogs, okay? Because if you don't use analogs, then the reaction goes on, and you cannot do crystallization. Okay? So these are the limitations for the conventional structural biology. Okay? So the recent development in structural biology, after having been able to do so many structures by different techniques, including the most recently developed cryo-EM techniques, now the field is moving to the time-dependent, or we call time-resolved X-ray crystallography or cryo-EM. Okay. Now, the current limitation of the time-resolved experiments is in the order of seconds or even milliseconds. Okay, but the biological reactions goes faster or much faster. Okay, chemical reactions even much faster, could go into the microsecond or even picosecond time scale. Okay. Now, cryo-EM has some advantage in that cryo-EM seems to be able to resolve multiple conformations. Okay, this is an advantage over NMR or X-ray. Okay, because if you have a mixture, then the process of solving the structure, you could resolve those mixtures for cryo-EM. And this is a recent review in 2022 that I have addressed the issue of dynamics in cryo-EM, but many other people in the field have also addressed this issue. So I'm going to tell you two stories today. The first one is on the cryo-EM, the use of cryo-EM to resolve conformational change in an induced field mechanism. And the second one will be the use of another emerging technology called X-ray free electron laser to resolve the reaction intermediates of a DNA repair process. So this is the enzyme for the first story. This enzyme is called keto acid reductase isomerase, or CARI, okay, as a simplification. And it goes through a two-step reaction, but it's a important enzyme in the amino acid biosynthesis for isoleucine, leucine, and valine. But don't worry about what it does. Okay, for now, let's focus on the structure. So this was the first structure we have solved in 2019 in Academia Sinica with our new instrument. So this is a dodecamer or 12mer. It is a hexamer of dimer. So there are 6 dimers. So this is a dimer. Okay, and this is a monomer. Okay. And the resolution, even though this is not the highest and most advanced instrument yet, it's 200 kilovolts, but we were able to resolve the side chain, amino acid side chains, even a metal ion such as magnesium at the active site. Okay. And then we were able to do the complex with the substrate. Okay, this is the analog of a substrate. Yeah. And then, in addition, the complex of not only the metal, the substrate, and also the coenzyme. Okay. And this is now, in a few years, just a few years, this is becoming quite routine now in the field. Okay. Every structural biologist, including many in the School of Life Sciences here, are working using this technique to solve many important biological molecules. And so this was published, as I said, during the pandemic. Well, that is not the end of the story. That's just the background. So from this work, we found something strange or interesting. Okay, that is this enzyme, but from different species. In the past has been studied by X-ray crystallography, the conventional one, where people use the protein from different species to do structures under different conditions, just like what I said. And then they have found a substrate-induced conformational change. Okay, this is the green structure is this enzyme CARI with 2 magnesium ion, and the red structure is, in addition, has coenzyme present. So this coenzyme presence induced a change of the conformation at this region. Okay, and this is very common in structural biology or in enzymology. That is, the induced fit mechanism means binding of a ligand can induce the protein to change conformation. And that is very good. But in our case, we did not see such an induced fit mechanism because our two structures under the corresponding conditions are almost identical. We didn't see this. Okay. So we started to think about the reason. And I think of two possible reasons. One is maybe the reason they observe this conformational change is because the two proteins come from different species. But the other reason is temperature effect. That is, even though cryo-EM is so popular, many people are working on that. One thing that most people have not told you is when they prepare the cryo-EM sample, they prepare it at 4 degrees. Okay. Well, cryo-EM by definition is performed at minus 180 degrees cryo condition, but the sample was prepared starting with a liquid and in a solution, and this solution is kept at a temperature that's for the purpose of stability or for the readiness for the freezing. They were kept at 4 degrees. But at 4 degrees, most proteins are inactive. Okay. So we thought maybe that's the reason. Okay. So we decided to do a time-dependent structure. Now, time-dependent structure seems to be, we look into the literature, actually no one has done it. Okay. So in the area of NMR, okay, variable temperature study has been done frequently, but not for solving the structure. Okay, you could do a particular NMR experiment to look at a specific region of the protein for temperature changes, but not for solving the entire structure. For the X-ray, there has been temperature-dependent study for the temperature of data collection. That was done. This is Greg Pesco. Okay, since 1979. So what they have done is that, okay, X-ray often data is collected at cryo temperature also, but some of them are done at temperature, at higher temperature, and they did that kind of study to look at dynamics. That's different from what we are interested here. Okay. Now, in X-ray, the question that corresponds to what we are asking is whether crystallization at different temperature has been done for comparison. And we only found one paper that is crystallization was done at 4 to 50 degrees, but there was no difference, no effect. But the crystallization is dependent on the crystal packing force. Okay, it's another important factor. Okay, now cryo-EM, what I just mentioned, is done at 4 degrees. FU cases were done at minus 20 degrees. Okay, so anyway, we decided to do 4 to 70 degrees at 6 temperatures. So we did that. We have to do it for two samples. One sample is without the NADH ligand. The other sample is with the NADH ligand and the inhibitor. So this is uninduced, this is induced. We should see an induced fit mechanism at different temperatures if our prediction is right. So the two conditions, one is, we call it A series, okay? Now, 4 means 4 degrees, 70 means 70 degrees. So this is B is the ternary complex that has ligands, 4 to 70 degrees. Now, we compare structures at 4 degrees between the two complexes, A4 and B4. So this is the A4 and B4. And as you could see, and this is the same as what you see in the earlier slide, that there is almost no conformational change at 4 degrees. Okay. But if you compare the two structures at 70 degrees, so you see conformational changes. So this is a conformational change, okay, pink color and the red color. The degree of change, we define it by the rotation angle. So this is rotation angle of about 15 degrees. Okay. And this is in between. This is if you stay at complex 4. So even for the same complex, if you change the temperature, there is a small change of conformation. So protein structure is dependent on the temperature. Okay, this is very important. Even though in the almost the entire literature, when people report protein structure, no one report temperature. Okay. So then, since this one has a conformational change, now we ask what is the intermediate. Okay, so most structural biologists consider a conformational change a two-step process. Okay, you go from this conformation to next conformation. What happened in between? Actually, we don't know, or we don't care. Okay, but it is important. Okay, so what we do is we could not detect the intermediates here unless we do a time-resolved process experiments, and this could be very fast. This could be in milliseconds or even faster. But what we could do is, since at 4 degrees, the conformation change has not happened yet, and we know at 70 degrees it happened. So what we do is we simply look at the temperatures in between. Okay. Okay. So we collected those data anyway. We look at those data, and so this is the comparison between 4 degrees and 70 degrees, and each structure here is a superimposed imposition between two temperatures, 4 degrees and 25 degrees, 4 degrees and 36 degrees. And you can see there is almost no change up to 50 degrees, but the conformational change started to occur at 55, and then it became more prominent at 70 degrees here. Okay, look at the blue color and the red color here. Okay, so the conformational change occurs at almost close to the highest temperature, 70 degrees. However, if we look at the active site, this is it's the same thing, just you zoom into the active site here. You zoom in from here and look at the details here. You could see that this is a substrate analog. Okay, at 25 degrees, the substrate already changes the conformation of the substrate, or the binding of the substrate. Okay, from the blue to the green color. Okay. And then at 36 degrees, it further changes. Okay. And then at 50 degrees, it no longer changes. Okay. In other words, now we know how the conformational change happens. It started at the center where the substrate binds, and it changes first, and then the global change occur at the last, and in between is if we look at the area of the coenzyme binding, NADH. NADH changes occurs in between the two extremes. Okay, this is a little bit harder to see the details here, but the trend is clear. So this is a summary of this study. That is, the conformational change now could be dissected into microscopic steps. You could do as many steps as you want. You could do, we change temperature. We only have to do 60 temperatures. You could do 60 temperatures if you like. I'm sure you will get detailed, more detailed information. So the summary is the observation of the process of a ligand-induced conformational change. And then it is important to solve cryo-EM structures at functional temperature. So this is the first story about the dynamics of a structure or structural change. And the second story is more of a different technology. In a way, this is the theory is the same as X-ray crystallography, conventional X-ray crystallography. It is the same, but the instrument is a bit different. Okay. So conventional crystallography uses synchrotron radiation, which is not a laser, okay, which is a very strong radiation. That's good, but which is ordinary light. So the difference here is it's also X-ray in the same wavelength region, but it is a laser. Okay. So the property of a laser is it has high peak intensity and it has short pulse. Okay, so you could do it very focused way. Okay. The pulse duration is very short. Okay, 10 femtosecond. Femtosecond is very short time. Okay. And it has two main advantages. So one is time resolved, as I explained. The other is the damage-free crystallography. Actually, the conventional synchrotron radiation could damage your crystal. The same thing with the laser, free electron laser. Now, this just want to explain to you where it could be done. There are 5 different places in the world you could do it. Closest place to us, one is in Japan. This is Sakura. So this is a conventional X-ray. This is the free electron laser. And then this is in Switzerland. Okay, we went to both places during the last 2 or 3 years. Well, we was disrupted in the first year of the pandemic, but then, then subsequently we continued. And this is what I say. This theory already was published very early. That under what condition, what timing, the damage to the crystal occurs, and femtosecond, 10 femtosecond is at the condition where the damage has not occurred yet. So in this technology, what you do is you use many crystals. So conventional crystallography use one single large crystal, and you collect all the data on this crystal, and some damage occurs in between. But in this XFEL, you actually use many thousands of microcrystals, and the crystals is shoot through as a stream, and then the X-ray beam goes through it. So every crystal is hit once and then done. Okay, it's no longer used. Again, the next will be the next crystal. And the reason you could do time resolved is, particularly for the light triggered reaction, okay, photo-induced reaction. This is most useful because of the fast time scale. So there are actually 2 different lasers. One is called the pump laser, which, for example, in this case, is the blue light that you hit the sample to induce the reaction, and then after, okay, this is the pump laser. Then after a delay of time, you shoot the X-ray laser and then collect data. And you could change the time T1, T2, and this T1, T2, because the pump laser is only 10 femtoseconds, so you could have a femtosecond resolution. So actually different facilities could have different resolutions. So in Switzerland's facility, the pump laser is a femtosecond laser, so we could have very high resolution, picosecond to nanosecond. In Sakura, Japan facility, the pump laser has a nanosecond, so we could only do the nanosecond to milliseconds. So these are all details. Now about how to solve the structure. So So since we are monitoring the changes or the reactions in very short time period, so you could imagine the changes will be small. Now, if you do X-ray crystallography or any structure, you know there is a limitation of your resolution. So when you show the students this is a nice structure, but you don't tell students this is only a model, structural model. Okay, every structure has some uncertainty. Okay, now, so when this time difference becomes smaller and smaller, then of course the demand for resolution becomes higher and higher. But that's okay. That's not a problem because here we are focusing on the difference between two times. So the difference is very reliable. So even though you saw one structure, you may have some uncertainty, but if you compare two structures, then you compare the difference in the map directly. That's reliable. So, and then of course, by going through the details of software analysis, which I'm not going to go through, but okay, so that's the methodology. And then the question would be then which problem are you working on? So the enzyme we work on is photodiase. Okay, this is the enzyme that repairs the thymine dimer of the damaged DNA. Now, okay, this slide looks complicated, I know, but if you are not in the field, but this is, let me explain to you, it's not complicated. Okay, so this region is the dimer. So this is TT. If you have two thymines in DNA next to each other, that's TT. Then under UV light, it forms a dimer, and that causes DNA damage. And this DNA damage is it occurs to plants, to organisms very, very often. But DNA damage is okay, but it needs to be repaired. And this enzyme is responsible for repairing this DNA, damaged DNA. But how the enzyme works, the enzyme needs the help of a coenzyme. This coenzyme is FAD. Okay, that's very common in biochemistry. But this coenzyme FAD has to be in the reduced form. So the coenzyme has to be reduced from oxidized form to reduced form. And this reduction process depends on electron transfer. So anyone you take biochemistry, you know FAD to FADH or FADH2. It's a 2-electron process. Okay, 2 single electron transfer process. 1 single electron in between, there's a protonation and the second single electron. Okay. So this is just an illustration of what I just explained. So now FAD, so this enzyme has been studied extensively previously by different technique, by spectroscopy methods, not by X-ray. X-ray it's typical, limited, but it was done by spectroscopy because FAD is a very good chromophore. It has different colors, and then under different conditions, the color could change, and then you could use spectroscopy, UV, well, IR or Raman to do this kind of study. So then we use X-ray crystallography. Well, at first, we use conventional, well, not conventional, not time resolved. Okay, we look at the static situation first. Okay, when you look at this, there is FAD oxide, there's one electron transfer. We just first look at these static positions first. Then we look at something in between. Okay, in between we look at many time points. Okay, but here what you see is we first look at the 3 major states: FAD, H dot, that's after 1 electron transfer. This is ADH minus 2 electron transfer. And then you do a superposition, then you do a difference map. So the different colors you see here represent different maps between this structure and the first structure, the second first structure. And then this third one is the third structure and the first structure. So as you can see here, the differences occur mainly at the active site. Okay, that is, the two structures are actually superimposed very well, but in the active site you see the differences. So our job is to interpret what happened, okay, what details in the active site here. Okay. So this is the expansion of this this region. You expand it, and then you do subtraction. Well, it's a subtraction. Then you try to analyze these data for their differences, and then you obtain the data, the changes in the detailed structures. Okay, it's like between this nitrogen from protein and this nitrogen on the coenzyme, how much the difference has changed. If you compare this one, it's 3.3 angstrom. If you compare it, look at it at the beginning, it is 4.8, and it's 3.9 here. Okay, so you deduce these detailed parameters. As an illustration, I'm showing that with one structure, but in reality, okay, before we get to that, this is just a summary of the example. Okay, with the comparison of the one structure here, the beginning and end structure, you could see that the difference is very small here. Okay, the two structures, the difference is very small. But in between, something could happen. Okay, in between, there could be larger differences. Okay, so this is a time-dependent series. Okay, so you don't have to look at the details here because it's impossible. But this is just to tell you that you could go from nanoseconds. I think the first one is 10 nanoseconds here, 10 ns, 10 nanoseconds here, 50 nanoseconds, 250 nanoseconds, 1 microseconds, and then up to 5 milliseconds. So this is what happened after transfer of electron from time 0 to 5 milliseconds, and this is a plot of a log scale. And as you could see here, at 10 nanoseconds, there is very little things happen. Okay, we are looking at the changes in dihedral angles here, but whatever it is, we have many parameters we could plot, but you just compare this 10 nanoseconds and the 10 microseconds. So 10 microseconds seems to be the peak of the maximum of this process. And then after that, it reduces back, reduces back. That's how the experiment was designed. And then you look at another parameter from the same data set. It tells you the same thing, that nothing has happened yet at 10 nanoseconds, but it goes through a process and reaches maximum at 10 microseconds. So this is the data that you could deduce. And then the question is, what does this data tell you? Okay, so the data tells us at 10 nanoseconds, the reaction probably has not happened yet. But if you look at another region of the protein, see the protein has many different parts, different amino acids. Okay, they are involved in doing different things. Okay, just like different instruments is playing different notes in a music. Okay, So here at 10 nanoseconds, we look at the other region. This is the histidine that has changed the conformation already. I mean, this is the overlay of two structures. So in one structure, histidine already turned to the other direction. So histidine has already changed conformation. So where is histidine? This histidine is near the electron transfer chain. There is a W tryptophan around here, 338. That was known to be involved in electron transfer from previous physical studies. Okay. So this is an example to tell us that, yes, at 10 nanoseconds, we see this change that could be attributed to electron transfer. So the electron has to transfer from the protein or from the light to the protein, and then to the FAD, and then FAD then undergoes a change of conformation as shown in the previous slide. Okay. So in the textbook, when you talk about a reduction reaction, usually one of the explanation could be a transfer of electron is a reduction reaction. Okay, but here we know that a transfer of electron is the beginning of the reduction reaction. Electron is transferred to the ligand, and then the ligand then has to digest that electron, accept that electron into the structure. Okay. Okay, so electron transfer is not equivalent to the reduction reaction. Okay, is that complicated enough? Now, some of you may think, why do you care about this? Well, this get into chemistry and physics. Okay, but you have to know, many physicists, they spend the whole life studying water molecules. Okay, how the ice is formed. Okay, so it's a different field. But I have to tell you that this is something the AI could not predict. Okay. AI is not there yet. Okay. Also, the reason we are not so successful yet in the drug discovery or drug design is because we don't know the protein, the enzyme, in sufficient quantification, in sufficient detail. Everyone thinks we know proteins, enzymes. Everyone thinks it's all done. Okay, AI could tell you. Okay, but it's not true. Okay, I mean, you need, when something matters, it's in the scale of angstroms. When something shifted by 1 angstrom or half of an angstrom, something dramatic happens. Okay, that's so. The next step is proton transfer. Okay, this proton. Okay, I mean, I just talked about this the first electron transfer, and then this needs to be protonated. Now, to look at how the difference between this transfer, proton transfer, what all we have to do is to compare this structure with this structure. Okay, so by comparing it with these two structures, then we know what happened. We know which amino acids are involved in this proton transfer. And then I'm not going to go through the details with you again. And then the next thing is we need to do the next step of electron transfer. And the next okay, this is about the proton transfer. The next step of electron transfer, again, we go through the same experiment as I explained above, but the changes are somewhat different. Okay, but they are similar. Okay, this is 300 nanosecond. You reach the maximum. Okay, but the point is, every process, every single step of the protein reaction, it could be dissected and interpreted. Now, This is now a summary of the entire process only for the preparation, the activation of the enzyme to repair the DNA. Okay, as I said, the enzyme needs to be converted from the oxidized state to the reduced state in order to do the DNA repair. So let's skip this and move to the repair. This is so this work was published last year in Nature Chemistry, and as you could see, this involves many people, but many of the people were simply involved in the synchrotron experiment, because that needs many people, including the staff in the synchrotron facilities to help. But the DNA repair should be more exciting. And okay, so as I mentioned, this has been studied extensively by physical chemists by using spectroscopy, and this is what has been proposed, okay, for how this cyclobutane ring is repaired. Okay, so again, it goes through electron transfer, and then electron get delocalized, and then the bond is broken, and another bond is broken, and then the repair is complete. Okay, that was a proposal, something you could not see, but something based on spectroscopic changes. So our goal, I have explained to you, we want to see more intermediate, we want to understand chemistry, because that is important. And here, okay, I don't know why the top one is not showing up, but that's not important. That's just, the top one is just a repeat of previous one. So here again, we did everything in the presence of DNA. Okay, the top row will be in the absence of DNA. Now in the presence of DNA, and then we want to interpret the differences in the active site. And the active site now has damaged DNA. So when DNA binds, the TT dimer, as you could see, the thymine dimer in the double-strand DNA actually flipped out of the DNA into the active site of the protein. This is the TT dimer here. Okay. But when we do the X-ray structure, as we know, we will see everything. Okay. But we may not be able to analyze everything because there's too many places to analyze. But what we have done here is we simply focus on one structure first, or one time point, and then we identify the differences, and then we will focus on these areas. We found these areas have differences. So then we will do a time resolved study, and when we do the analysis, we focus in on this area. Okay. So the lower right region, for example, this is the region when damaged DNA, the thymine base flip out of DNA, so you see some emptiness here, but otherwise this is part of DNA. Okay, so then this is the time resolved. Okay, and this is simply part of the active site, part of what I showed in the previous slide, and this is the damaged DNA. And this is 100 picoseconds. Okay, picosecond is you know how short it is. So here you don't see a change of this damaged DNA. You don't see a change elsewhere, but you see a change of arginine side chain moving from here to here. Okay, that means something. Okay, because this is 100 picoseconds. This is 250 picoseconds. That's the same thing. I'm just focusing on. I'm singling out this region for you to see. The damaged DNA, nothing has happened yet. Okay. But the arginine has already moved, and this is the region of FAD. It has also changed at here at 250 picosecond. So this means electron transfer has occurred, because that's what we learned from the previous study, that when FAD changes conformation, that signals some electron transfer. And then also this substrate probably is charged or negatively charged now because of the electron transfer. Therefore, the arginine is moving here to neutralize it. Okay. And then at 650 picosecond, something started to happen. Okay, this C5 prime bond is broken because this distance increases. Okay. This bond is still intact, and then here both bonds are broken. That means the cyclobutane ring is entirely open. Okay, the two bases, two thymine are still there, but their distance increases by an angstrom or so. This is what I mean. When something matters, it is in the scale of angstroms. Okay, sub-nanometers. Okay. Now that's 1 nanosecond. So we think the reaction is complete in 1 nanosecond. After 1 nanosecond, based on similar analysis, we could see that the electrons from the substrate is returned back to the coenzyme. That occurs at 2 or 3 nanoseconds. And then between a couple of nanoseconds up to 500 nanoseconds, that's a long time. Okay, 500 nanoseconds is a long time compared to 1 or 2 nanoseconds. During this process, we don't see much happens. Okay, because these are all similar. The maps are all different maps, and they look similar. Now this is just from different angles. If you see small differences, and the coenzyme also look differences. So what happened between 6 and 500 nanoseconds? Okay, I'm going to skip this one. Okay, we look carefully on the enzyme to see what happened between 1 nanoseconds here and 500 nanoseconds here. Okay. Now you have to believe my word because this is very complicated. But if you look at this region, okay, there are 5 small balls. These are 5 water molecules. Okay. Now there are different colors because it's a superposition of 2 structures. Now these 5 are superposed well, just like the beginning, because the beginning 100 picoseconds. Okay. But once the reaction happens, okay, like in nanoseconds, like in 1 nanosecond, these balls are separated, right? Okay. And there are some other things like arginine. Okay, arginine, they are different. So what happened is, at the beginning, when the enzyme is resting, there's a structure. Okay, and then the active site residues, in this case, the water is involved, or arginine is involved. Then they started to perform functions and perform chemistry. And chemistry is complete in 1 nanosecond. But at this time, the enzyme is not recovered yet. Okay, the active site moves. I mean, the residues moved, or water is disorganized. And then between 1 nanosecond and 500 nanoseconds, the enzyme recovers. Okay. 500 nanoseconds is a long time, but it's a short time. Okay, it's less than 1 millisecond. So now the same thing, this is exactly the same picture like the previous slide, except we are looking at different regions of the enzyme. Okay. So at 500 nanosecond, what we see is the enzyme, okay, or the entire region, this region still empty. Okay. So even though at 500 seconds, I say the enzyme active site recovers, but the DNA has not returned yet. Okay, the thymine is still out there, but after 500 nanoseconds, so the product release started at 500 nanoseconds. So this is after 5 nanoseconds. This is 25 microseconds. Okay, this is 200 microseconds. Here you could see very cloudiness. Actually, you could see some thymine. This is a thymine here. This is another thymine here. They return to pair with the DNA, fill up this gap. And this one is the same as this one, just not showing omitting the density map, just so you could see that these thymines are returned. Okay. Okay. So this is, so the previous slide shows some active site region, which from 1 nanosecond to 500 nanoseconds, active site recovers. This is also active site, but this is closer to the DNA part. So the repair thymine returns to DNA to reform double-stranded DNA at about 100 to 200 microsecond range. At that time, the product DNA is supposed to be released, okay, but not yet. I think at that point, DNA still hangs around. Okay, it's just about the time to release. So at that time, you see a lot of different densities. That means there's dynamics there. Okay. So this is the complete process. That is the CPT repair. This occurs at the first nanosecond. I mean, this is a log scale time. This one is 100. So, and then the active site recovers. Then TT returns double-strand DNA, and after that there will be residues of product, and this will give you a picture of the process. Okay, so different regions of the protein is shown, and the time is shown. Okay. Oops. So now we could hear the music. Okay, you want to keep going or you want to stop? This is from Chicago Symphony. This will take 90 minutes. Okay, let's stop here. I may have spoken too fast because I was afraid to bore you too much with the details, but we could have time for discussion. Okay. Yeah.
Okay. Thanks, Prof Tsai, for his eye-opening, talk on how you look at, using structural biology, various things of structural biology, look at how protein functions at real time. Okay, I guess the floor is now open for questions and Q&A. Any questions from the audience? If I may, I may want to get the ball rolling for asking the first question. So in your first part of the work, you use the cryo-EM single particle analysis to look at the enzyme, the enzyme structural change from 4 to 70, right? So apparently it goes through different sub-state, right? From here, and then to the final state at 70. But in a sense, because temperature is the thermodynamic,
right.
It's an ensemble, right? Probably. So when you do single particle, wouldn't you expect that a particular population would shift its relative, proportion by changing the temperature, rather than you see it move gradually from one plate to another? So you see it.
Yeah, this is a very good question, because in structural biology, no matter what you do, you see a population. Everything is a distribution. The question is, how much do they distribute? Now, the reason we don't see the distribution, we don't see details, if it's because we don't have the sufficient resolution, or we don't have the sufficient time resolution. Now, even for any chemical reaction, for sub-steps, it is still, as long as you have a collection of molecules, it's going to be a distribution. Now, the conformational change is a kinetic event, okay, and that's always a distribution. Now, what we do is we convert a kinetic problem into a thermodynamic situation. Well, we use temperature to control the step or the pace of change. Now, temperature is a thermodynamic effect, and you are absolutely right. All the molecules will merge or consolidate at that temperature, determined by that temperature. It's still a collection, maybe a different collection, but obviously it's a focused collection. Obviously, when you go from one temperature to the next, the overall is different. Even if it's no matter how broad the distribution is, you see a difference of the main species.
If I try to make sure I understand.
Yeah.
So I think I use the MR concept, right?
Yeah.
You have you put a very simple model of 2 states. Okay, and 4 is this state.
Yeah.
It could be more than 2 states, but for imagination, it's easier to think of the 2 states, and 70 is the 2 states.
Yeah.
By changing the temperature, you're moving the equilibrium. Basically, the weighted population function of temperature.
Yeah.
So basically, what you see is still an ensemble average, but the mean of the ensemble average are moving from 4 to 7, because the relative ratio, the 4 state at 4 degrees state and 70 degrees state is different.
Yes.
Of course, we can have more than 3, but the mathematics will be much more complicated.
Yeah.
Oh, I see that's quite brilliant.
Yeah.
I think that's
But in NMR, of course, people trust that once you do a temperature, if the peak shifted.
You see the
peak shifts. That's right. And people do interpret that as a structural change.
Oh, yeah, that's true. So okay.
So it's a very we're getting deep into the problems of kinetics and thermodynamics and molecules.
The temperature as a surrogate.
That's right.
The equilibrium populations of 2 different states.
Yeah.
That's very interesting.
But the importance is it's not a two state. That is,
It has to be.
we see change, we see differences in the different region of the molecule. See,
Yeah, yeah, yeah.
so the change at the active site is different from the change in the remote site. At least the time dependence or the temperature dependence is different.
But the same logic goes to a multi-state, right?
Yeah.
You can imagine at any given temperature, you've got multiple state, and every have a single population. But you change the temperature, you change the distributions of the substrates.
Yeah.
And
But
then what you're measuring is actually the so like ensemble average state.
yeah.
So you still see the movement. From
But it's not like if you stand here. If I move here, it's not like I move this way. No. But no, it's like if I move, maybe my hand is moving first, and then my leg is moving first. Maybe then my body is moving. It's not static.
No.
So this tells you, his hand is moving first, and then his leg is moving, then his body is moving. Not like many people think. Well, we just quantumly, tunnelingly.
So may I say that using also very related to NMR concept, right?
Yeah.
So it means that what you're seeing is time and ensemble average, right?
Yeah.
But I assume that because using cryo-EM, the speed you mix the sample, the conformational change must be faster.
Yeah.
Right? Faster, so that you don't see two different states.
Yeah.
If you frozen it fast enough, you see two distinct populations.
Yeah. But there is a difference between NMR and cryo-EM. That is, their dependence on dynamics is different. Okay, so in cryo-EM, if you move, okay, suppose this is an ensemble. If the range of ensemble is too large, too broad, you would not see the map. So it has to be fairly focused. But anyway, every technique is different.
So we are actually getting into a lot of biophysics. That's why we need to study physics and make touch upon back and statistical mechanics, right?
That's right.
That type of thing.
That's right. I mean, these kind of concepts are not hard. If you are students here, I mean, there are how many of you are students here?
There has to be student, right?
Yeah. I mean, this concept is not difficult. You will get it from your physical chemistry class. If you get it right, you could use it the whole lifetime, because when you think about things, you think about things in this way, in a deeper way, or you could, this is just basics, kinetics and thermodynamics.
Yeah. That's actually a very good book, authored by John Perrion, which is one of the very famous structural biologists, called The Molecular of Life.
Yeah.
For those interested, you can read that.
Yeah.
Okay, maybe I pass the time to audience. Any more questions from the audience? Any from the web? Wait.
Professor Tsai, so I must first say that I'm not a structural biologist, but I'm intrigued by your analogy of your research to about physicists who study, or some physicists who just study modern molecules. I'm just wondering, so water is so important in triggering or in facilitating enzymatic reactions. And given that you're looking at cryo-EM, and cryo-EM would be needing like -70 degrees Celsius of preparation condition. Is it possible, instead of changing temperature, can you actually reducing the number of modern molecules sample to limit the rate of enzymatic reaction so that you can make use of such limited modern molecule environment, freeze down the individual substance of it, so that you can have a sample of the whole reaction?
Well, it is certainly possible. That is, the enzyme reaction is indeed dependent on the medium. It's like if you add glycerol to your water, your buffer, then you could slow down some reactions. Okay, but not every step. So so enzyme reaction involves chemical steps and conformational change steps. The conformational change step is more likely to be affected by the medium. Okay, and the chemistry is less likely. Okay, but that's the medium effect. Okay, viscosity effect. Okay, but on the other hand, there are some water molecules in the active site which are involved in the reaction or in the binding with between enzyme and ligand, which are quite independent of the medium effect. Okay, therefore, even in some crystal situation, some reaction happens at crystal condition. Okay, so it is possible. There are different kinds. That's why I say there are people who are interested in studying water. Even water is difficult to study. Yeah.
Okay, easy. You increase the temperatures.
Yeah.
And then find some induced heat.
Yeah.
But at the same time, I suppose the proteins begin to delay.
Yeah. No.
You eliminate it.
No, by the way, I forgot to mention this enzyme. This enzyme is thermophilic enzyme. Yeah. So I should have said that, yes, but even regardless of this one, okay, the difference between 4 degrees and 37 degrees are different. So actually, we have another work that I have not talked about. We look at the spike protein of coronavirus. Look at 4 degrees and 37 degrees, and there are differences. Even the conformation is different. The RBD,
FIB is the one that
the RBD conformation will be different, right? So dynamics is a difficult issue. That is important, but have not been studied well previously due to the limitation of technology and methods. But I think the field is shifting that way.
I guess the AlphaGo will have a hard time showing what you show, right?
That's right. So
we still keep our
so
job.
that's right. So
this, yeah.
so we should not be afraid of losing our job to
something else.
because you need to do something that AlphaGo could not do. Okay, if you are doing something that AlphaGo could do, then you may as well just do AlphaGo.
Yeah, I also ask myself if ChatGPT can give an answer better than you guys.
That's right.
Then you're all out of job. Okay, more questions from the audience. Any? Okay, gentlemen.
I'm interested in the second story about the photolysis.
Yeah.
So using the time resolve X-file.
Yeah.
So you can see a lot of these images. So if let's say we have thousands of microcrystals.
Yeah.
So what's the next technical challenge, okay, to get all these intermediate resolved?
Okay, that's certainly a very good question. So conformational change is one thing. I think you just asked about conformational change. There could be an ensemble or a shape of ensemble. Now, in the chemical reaction, so at least in the first 1 nanosecond, we are looking at chemical reaction. Now we are looking at sub-nanoseconds. So there is also a possibility of an ensemble. So if you have 1,000 molecules, they don't synchronize exactly. If you look at single molecule situation, they are slightly different. But you will see a majority. That is, when you see a distance of 1 angstrom and 1.2 angstrom, then if the 1.2 angstrom I don't remember what is the number, but or 2.2 angstrom. You know where you have to interpret. You have to know where the majority of the bond is broken. Okay. So in other words, to be careful, we still have to call our intermediate the predominant species, not the single species. Even though when we talk about lectures in general, we say that's a single species, but in reality, it's still an ensemble. And in some cases, you could see a superpose of two conformations. That's that's not very uncommon in structural biology, that you see two conformers in the same data set. And that could happen also in X-FIL.
One question from the audience. I have one more question.
Sure.
If we think out of the base from the double string here,
Yeah.
for repair or modification, it's
Yeah.
quite a common phenomenon. So I just wonder if your experiment has also investigated how these phase are being rebuilt.
Well, we know what it looks like when it is ripped out. When it's been flipped back, we have some intermediate. I didn't show that again because of too much detail, but it started with this, and then it goes like this, and then it goes like this. There are intermediates. So one base comes first. We caught that intermediate. We are arguing with a reviewer about this now. Reviewer thinks it should be a two-step process, but we say we observe this intermediate.
It seems to be that, for example, if you methylate a cytosine,
Yeah.
the cytosine needs to break up.
Okay.
Then you need to have some bond breaking before you can push.
That's right.
You can break up quite easily without any sort of.
Well, yeah, but every enzyme is designed to do what it's supposed to do. So in the enzyme involving the methyl cytosine, it will have its own active site that will facilitate that. So here, the enzyme have active site residue to stabilize the damaged DNA. But when DNA is no longer damaged, this repair is done, then the binding becomes unfavorable. Then it has to come back.
Okay.
Yeah.
I can ask the second one then. Okay. So maybe follow up the questions on flipping out and feedback of the family diamond. So I'm sure you must have seen what triggered it, right? Because you've seen all the progress.
Yeah.
There must be something that stabilized the conformation when it flipped out. So I see a lot of subtle changes, on the active side.
Yeah.
So what induced it? Induced the diamond to flip back?
Well, honestly, we don't know. We don't know.
Okay.
I mean, what we know is only what we have analyzed.
Yeah.
And then obviously Watson Crick base pairing is when it flips back, it's meeting the partner. T will be meeting A there. That could be a driving force. But the whole thing, I mean, how do you start from the beginning of the music to the end of the music? Whether there is a conductor there, we don't know. Yeah.
If I may, I ask the final question.
Sure.
Okay. I guess you can trace the whole reaction. Is it because the reaction is induced by light? I mean, by
Yeah.
Otherwise, for other reactions, it's maybe not that easy for the other system.
That's right. That's right. So the most studied system by this technique is rhodopsin. Okay,
Yeah,
that's light. And then the photo systems. You have seen many papers in Science Nature about these papers. Now there are more and more papers that use XFEL to study a non-photo reaction, but they solve other problems. Okay, but they are not, those papers are not published in Science or Nature, but they are in other good journals also. That is, they don't do time resolve, but they do use there's still advantage of, it's an undamaged condition and maybe better resolution. And then there is also a group of people who are designing caged substrate, that is using something photoreaction to trigger the reaction. So I mean, that's the future. So I would say that there is two emerging techniques for in structural biology: cryo-EM and XFEL. I think they are equally important for the next 10 years.
Yeah, that's good to hear. Okay.
Yeah. By the way, the first author of this work has become a professor in National Taiwan University because of this work. So the last part, the repair part, is still being reviewed, not published yet. But this is so the color ones. So this is the international consortium, but this is done by TPP. So all of even though there is a lot of international names, the pink color names are all done in IBC, in Academia Sinica. And but I'm just about to finish my job, and then the first author has moved to National Taiwan University. But at least this work will continue in Taiwan.
Yeah, great. Yeah. Okay. I guess that's the end of the second lecture from Professor Tsai. Just a little bit advertisement. He will give a talk at Science Museum this Saturday at 2:30. Okay.